This video is part 2 for subtopic 3.5, Genetic Modification and Biotechnology. The understandings, applications, and skills that we will cover in this video will be related to that of cloning plants and animals. So clones are genetically identical organisms or a group of cells derived from a single original parent cell. Now, organisms that reproduce asexually will produce genetically identical clones. Additionally, mechanisms exist whereby sexually reproducing organisms can produce clones. For example, identical twins are clones. So there are natural cloning methods as well as artificial cloning methods. So with those natural cloning methods, many species can reproduce asexually and hence possess natural methods of cloning. All bacteria, the majority of fungi, and many species of protists reproduce asexually to produce genetic clones. While most plants reproduce sexually, they also possess methods of asexual reproduction, which is called vegetative propagation. Certain animal species can also reproduce asexually through a variety of different mechanisms. Komodo dragons are one example of a species that can reproduce asexually if the female is isolated from the rest of the population. With animal cloning, it is a lot less common than plants. A common technique for cloning is called budding. This is where cells split off from the parent organism, generating a smaller daughter organism, which eventually will be separate from the parent. This method of cloning occurs in Hydra, but is also common to many species of yeast. In plant cloning, plants have the capacity for vegetative propagation, whereby small pieces can be induced to grow independently. Garlic and onion bulbs are modified plant leaves. All the bulbs in a group are genetically identical, so they are clones. Basically, you can take a bulb from a garlic, plant that, and it will grow into a plant. Now you can have underground stems, for example, potato tubers, that can form new plants which are genetically identical to the parent plant. Now, certain plants can form horizontal stems called runners that grow and develop into clones. So strawberry plants reproduce in this manner, for example. So in that example of runners, here we have our plant, and they will produce like this little offshoot here that can root itself at a distance from that parent plant and grow into a plant that is genetically identical to the parent. And that plant itself can also produce runners itself. So in this way, the plant can propagate itself. One of the things that you will be asked to do, not necessarily on the exam, but expected to do just through the IB curriculum, is to investigate factors that affect rootings of stem cuttings. So a stem cutting is a separated portion of a plant stem that can regrow into a new independent clone via vegetative propagation. So basically you can take a stem or a branch or a cutting from a plant, place that into a bit of water, and it should start to create roots, which can eventually be planted, which will give rise to a new plant. Now, all stems possess nodes from which a leaf, branch, or aerial root may grow. The region between these nodes are called internodes. Stem cuttings are typically placed in soil with the lower nodes covered and the upper nodes exposed. Stem cutting is a common method employed to rapidly propagate plant species, including things like sugarcane, grapes, and roses, for example. So grapevines generally will be propagated through stem cuttings and will often be grafted onto other types of plants to make those grapevines a lot more hardy and resistant to different conditions. Now, there are a variety of factors that will influence successful rooting of a stem cutting, which includes the cutting position, whether the cutting occurs above or below a node, as well as the relative proximity of the cut to the node. The length of the cutting, including how many nodes remain on the cutting, as well as the growth medium, whether left in soil, water, potting mix, compost, or open air, and the use and concentration of growth hormones. Now, temperature conditions can also affect stem cuttings as well. So most cuttings will grow optimally at temperatures common to spring and summer, and the availability of water will affect how well that cutting will grow, as well as other environmental conditions, which include pH of the soil and light exposure as well. So these are all the different factors that can affect stem cuttings and affect rooting that come from stem cuttings. With cloning animals now, so it's really all about cloning animal embryos. So at early stages of development, animal embryos are pluripotent, and it is theoretically possible for the embryo to divide into two or more identical parts. However, there is little interest in this method of cloning since it's not possible to assess whether the individual has desirable traits. So in terms of cloning adult animals, cloning using mammalian cells is a lot more difficult. It's a very complicated process that doesn't have a high success ratio. Now, the first mammal cloned was a sheep named Dolly, and this was done in 1996. Now, Dolly the sheep was the first clone after 277 attempts. So this was the first viable sheep that had been born after 277 attempts. She was produced by what is called a somatic cell nuclear transfer. Basically, they took a somatic cell, which is a non-gamete cell, so basically a cell from the body, and they took the nucleus out, or they transferred the nucleus, sorry, of that cell to another cell. So for Dolly the sheep now, adult cells were taken from the udder of the donor sheep and cultured in a lab for 6 days, and this made the genes inactive. An unfertilized egg was then removed from a different adult female sheep, and the nucleus from that egg was then removed. One of the cultured cells from the donor sheep was placed next to the enucleated egg, the egg that had its nucleus removed. A small electric pulse was used to fuse the two cells together, and from that, about 10% of the fused cells began dividing to form an embryo. That embryo was then injected into a surrogate mother, and basically only about 1 out of every 29 embryos implanted developed. And this ended up being Dolly. That one ended up being Dolly. Now, diagrammatically, what this looks like, because there's a lot of steps involved in there, and it all sounds very simple, but it's all very complicated, requiring very specialized equipment to do all this. So, when cloning Dolly the sheep, so if this is Dolly here, a cell was taken from Dolly's udder, and this is considered the donor nucleus. So that cell has, that full set of DNA that was cultured, which allowed the genes to become inactive. Now, from a separate sheep, an egg cell was obtained, and that nucleus, this part here, was removed from that egg cell, so it was enucleated. The the two cells, so the egg cell and the donor nucleus, were fused together using that electric shock. So now that nucleus from that donor cell is now in the egg cell itself. And so this egg cell will now continue to divide, or should start undergoing mitosis, which would develop into an embryo. And all of these cells now contains the genetic material that originated from Dolly the sheep. And so as that embryo continues to divide and the cells continue to multiply, eventually that embryo is going to be placed into the uterus of a foster mother. And if things go well, that embryo should continue to divide and develop into a fetus, which then eventually gives rise to that lamb, which is a clone of that donor sheep, which is Dolly. So then you have your cloned lamb. So that is the end of this video for part 2 of 3.5.